Okay, so let me get this started here. Okay, so today we're going to talk about DNA, and specifically in this lecture, we're going to talk about DNA structure. And this starts our whole unit off of that has DNA structure, DNA replication, which we'll talk about next class, and also protein synthesis and RNA and how they are different and similar to that of DNA and DNA replication, and how that all comes together and works together for us to express our genetics. So, and we're going to kind of straddle that. So we're going to do DNA structure and replication for the 2 weeks before Christmas break, and then we're going to come back, review a little bit, and then start into RNA and protein synthesis when we get back from Christmas break. Okay. So, but today we're going to start with DNA structure, okay? And actually the DNA structure technology and the fact that we know that it is double helix is actually quite new, considering the fact that we've known that genetics is passed down from parent to offspring. We just didn't know how it was organized, right? And Watson and Crick, these two gentlemen here, and Rosalind Franklin. She was all three of these individuals and scientists, very brilliant, discovered the structure of DNA. Rosalind Franklin was specifically in charge of the X-ray crystallography, which is an older technology which allowed Watson and Crick to see, oh, this is a DNA is actually a double helix. And from there, they took that information and created what we know as the structure of DNA today. Now, Watson and Crick did win the Nobel Prize for that. However, Rosalind Franklin was not a part of that Nobel Prize winning. She was a woman in science. Back then, it was quite different than our society today, although we still struggle with that. And so she wasn't as highly respected as Watson and Crick due to the fact that she was a woman in science. So moving on, you don't really need to know any of that for your tests or your quizzes. However, it's a widely known thing that Watson and Crick discovered the structure of DNA. So, okay, so we're going to start with here is DNA, and DNA stands for deoxyribonucleic acid, okay? And remember, a nucleic acid, its monomer is a nucleotide. So nucleic acids are made of nucleotides, and nucleotides have 3 pieces that create a nucleotide. And that's number 1, your phosphate group, which is always depicted as a circle. Number 2, your pentose sugar, and for DNA, that sugar is called deoxyribose, okay? And number 3 is a nitrogen base. Now we have 4 different nitrogen bases. That's adenine, which we abbreviate by the letter A, thymine, which we abbreviate by the letter T, guanine, which we abbreviate by the letter G, and cytosine, which we abbreviate with the letter C. Okay, so we have 4 different nitrogen bases. Okay, notice if you look down here, adenine and guanine, they look very similar. Thymine and cytosine also look very similar. Okay, so keep that in mind as we continue on through our lecture today. So if we look at our DNA, and if we look at just one side or strand of our DNA, we can see that our phosphate and our sugar are repeating interchangeably. So we have a phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar. And that is what we call the sugar phosphate backbone, because it never changes, okay? Each one of us as humans and any organism that is living and has DNA has the same sugar phosphate backbone. What is different about our DNA is our bases and the sequence our bases are in. Okay, so that's super important to know. That will be on your EOC. It will be on your quiz and your 9 weeks test and your final. okay? Knowing what parts make up what of a nucleotide. Okay, so your sugar and your phosphate create that backbone that never changes. The only thing that changes in a nucleotide is your nitrogenous base. Okay, so let's look at this picture right here, and I want you to pause me for a second. And I want you to look at specifically the two sugar phosphate backbones here. What is different about them? So go ahead and pause me and then think about that, and then kind of tell yourself in your head and analyze this structure a little bit and see if you get it right. So if you thought about it for a second, if you notice that your phosphate group is up this way, okay, and then you have your sugar, and then you have phosphate and your sugar. Well, let's look at our backbone here. We have a sugar and then a phosphate, but then a sugar and then a phosphate. So we started with a sugar on the right side, but we started with a phosphate on the left side. Why is that? And is that important? Also notice that your bases meet in the middle and they are held together by a hydrogen bond, okay? We'll talk about bases and how they come together and what binds with what in just a moment. So if you said, hey, this one is right side up and this one is upside down, that's correct. And what we call that is anti-parallel. Okay, so anti-parallel means, the fancy way of saying that each side of your DNA molecule, okay, it is parallel, okay? However, one is the mirror image of the other, okay? We have to name that. So we say that one strand is 3' to 5', while the opposite side is 5' to 3'. Now I want you to write that down, and then we're going to go back here, okay? And I'm going to show you what that means and why we came up with these 3 and 5 and what these numbers mean, because it's important to know which strand is 3' to 5' and which strand is 5' to 3', because when we talk about DNA replication in our next class, okay, in our next lecture, you need to, certain enzymes can only go one direction. So one strand will be very easy to synthesize and create more DNA, whereas the other is going to be a little bit more complicated. So we need to learn how to identify our 3' and our 5' ends of our DNA strand. So let's go back here to this picture right here, okay? So let's just talk about organic chemistry. So this is a quick organic chemistry lesson, okay? So remember when we talked about biomolecules, we talked about how scientists are inherently lazy. We've talked about that a lot. But scientists are inherently lazy, and especially organic chemists, because they have to write a lot of carbons. So when two lines come together, like these two right here, where they come together, we assume that is a carbon if there's nothing else drawn in. So like for here, this one's an oxygen because it says it has an O. But these two right here, this one is definitely carbon, and this is a 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 carbon. Okay, so we have 5 carbons. So what we do is we number them, and we're going to start with this little tiny corner, okay, to the right of our oxygen and to the left of our base, okay, and that's going to be carbon number 1. The reason why we start with this one is because it is the most what we call chiral carbon. You don't need to know all that, but basically it has a lot of things attached to it. That's what that means, okay? So we're going to start with number 1. Then we're going to go clockwise. Number 2, 3, 4, and 5. So this right here, this 5th carbon is up. So this is our 5' end, okay? If we go over to this side, we're going to start here, right? Because this is the carbon that is to the is right next to our oxygen and also next to our base. So we're going to start here. 1, 2, 3, 4, 5. So the 3rd carbon right here is up. So this is our 3' end. Now, we know that these these are opposite of each other. So if this one's 5 prime, this side is 5 prime over here on the left, then the one over here on the right has to be 3 prime. We don't even have to count it. Okay? And therefore, if this one is 3 prime up here, the carbon that is closest to the end right here is 5 prime down here. And therefore, this one across down here would be 3 prime. Okay? So as long as you can figure out one, you can figure out the rest. Okay? So this strand right here on the left would be from 5 prime to 3 prime. And this strand over here on the right will be 3 prime to 5 prime. Now, what we also just saw in our DNA molecule is that the bases that are in the middle, remember your bases make up your rungs of your ladder, and what they create is a unique sequence that is unique to only you. We are all humans. Two organisms of the same species will share a lot of the same DNA, but it will never be exactly the same. Okay. And what changes that is the sequence of your bases. Okay. So those bases are held together by hydrogen bonds. Adenine will always bind with thymine, and they have two, AT2, they have 2 hydrogen bonds. Guanine will always bind with cytosine, and they have 3 hydrogen bonds. So I want you to ask yourself, which one is stronger, 2 hydrogen bonds or 3 hydrogen bonds? If you said 3, because that makes the most sense, right? More hydrogen bonding, the stronger the molecule. Yes, that's correct. So if you have 3 hydrogen bonds, that is stronger, okay? And I like to remember that A always binds with T because apples grow in a tree, okay? And G always binds with C because cars go in a garage, okay? And that's really all you have to remember as far as creating a complementary strand. So we call that A always binding with T, C always binding with G. We call that complementary base pairing. Okay, and Chargaff, he was a very logical scientist. Imagine that. And he said, well, if A always binds with T and C always binds with G, then I can count the number of A's or adenines in my sequence, and that should equal the number of thymines in my sequence. And same with cytosine and guanine, right? If I have 100 cytosines in my sequence, then I should also have 100 guanines within my sequence. Now, is it possible for that to not happen? Absolutely. That means that we have some kind of mutation somewhere. And we will talk more about that once we get into heavy genetics in the spring semester. So before I get into purines and pyrimidines, I'm going to go back a little bit to our second slide here. And I want us to look at this bottom picture down here. Okay, so notice your pyrimidines are thymine and cytosine. Okay, and your purines are adenine and guanine. Notice that these two groups, they look alike. They are grouped for a certain reason, right? So thymine and cytosine, I like to remember that they have a Y in them. Okay, so they are pyrimidines, which has a Y in it. Okay, notice they are only single ring structures, whereas purines, adenine and guanine, are double ring structures. Notice that a purine will always bind with a pyrimidine. So A will always bind with T, okay? And that creates 1, 2, 3, a total of 3 rings across, okay? And G will always bind with C, which again creates 1, 2, 3 rings across for our, if you think about DNA as a ladder, for the rungs of our ladder, which is important because if thymine bound with cytosine and adenine bound with guanine, our DNA molecule would not be even in the middle. It would be bigger in some places and smaller in others. And we know that that is not structurally sound. That is not a strong structure. And we need our DNA to be a strong structure because it is so important. And it basically tells your entire body and that specific cell what it's supposed to do. okay? And we'll talk about how it does that when we talk about protein synthesis, which is like brain explode emoji, cool. All right, so our purines are adenine and guanine. Remember, those are our double ring structures. And our pyrimidines are cytosine and thymine. Another way to remember your pyrimidines is cut pi. That U stands for uracil. We'll talk about that. Uracil is only found in RNA, and we'll talk about that when we talk more about RNA and all that good stuff. All right, so if you just take a moment, I want you to remember how we numbered our carbons, okay, and how we're going to label the different sides of our DNA molecule. So label the ends of this DNA molecule to the left. You go ahead and pause me, and then I'll give you the answers here. So A, notice our fifth carbon, 5' carbon is up. So the A is our 5' in, which means C has to be our 3' end. Very good. And therefore, if C is 3', then D must be 5'. And across, if D is 5', then we know that B is 3'. Okay? And here you can see how it is actually numbered and everything like that. All right, so Chargaff said, hey, if the number of A's must be the same as the number of T's and as well as G's and C's, right? However, he also said this, just because we're restricted on our complementary base pairing, meaning A will always bind with T, G will always bind with C, that doesn't restrict us on the combination of sequences we can have, okay? Think about this. You and me, we're both humans, right? We have some similar DNA, but we're not exactly the same, okay? We each have our own unique DNA sequence, okay? But our A's will always bind with our T's, both of us. Our G's will always bind with our C's, both of us. However, how many A's and T's we have within our sequence may be completely different for certain genes, okay? So it doesn't restrict our combination of what our sequence can be, okay? So that's super important to think about. So let's go ahead and review. Go ahead and think about what are the 3 parts of a nucleotide, what parts of a nucleotide make up the backbone of DNA, and what parts of the nucleotide make up the rungs of the DNA ladder. For number 3, the rungs are the steps that you actually step on a ladder. It also creates that sequence, that uniqueness, okay, of each and every one's individual DNA sequence, okay? So pause me and write down the answers that you think, and then I'll go over them. Okay, so what are the 3 parts of a nucleotide? Well, that is your sugar, and then for DNA, that's our deoxyribose sugar, phosphate, that sugar, phosphate, and then we have our base, and that base can be A, T, G, or C, okay? What parts of the nucleotide make up the backbone of DNA? Well, remember, the backbone is alternating phosphate, sugar, phosphate, sugar, phosphate, sugar, okay? So it's sugar phosphate is the backbone. And what parts of the nucleotide make up the rungs or your sequence, okay? And that is your nitrogen or nitrogenous base. That's your adenine, thymine, guanine, cytosine, ATGC, however you want to say it. That is the most important part of the nucleotide because that's what makes you you. That's what makes your sequence. That's what makes you unique. Now, a couple more questions. How are the bases held together when paired? How many hydrogen bonds does adenine and thymine have? How many does guanine and cytosine have? If the DNA double helix, one sequence is AGT, CGG, what is the sequence on the complementary strand? That's what that question is asking. It just sounds really complicated. Number 3, how do you identify the 3' and 5' ends of the complementary strands of DNA? And which DNA strand below would have the highest melting point? Okay, so go ahead and pause me and answer those. All right, so how are the bases held together when paired? Well, they're held together by hydrogen bonding, okay? And hydrogen bonding is a really cool special intermolecular force, which you'll learn about in chemistry, but it only happens between hydrogen and either oxygen, nitrogen, or sulfur sometimes. But it's mostly just hydrogen and oxygen or hydrogen and nitrogen. Okay, so adenine and thymine have two hydrogen bonds, and guanine and cytosine have 3. Okay, so if I gave you this sequence, AGT, CGG, then our complementary strand would be A binds with T because apples grow in a tree, G binds with C because cars go in the garage, T must bind with A, C binds with G, G binds with C, G binds with C. So our complementary strand should be T, C, A, G, C, C. Okay? And how do we identify the 3' and 5' ends? Remember, we're going to count those carbons, okay? And if our 5th carbon is up, that's our 5' end. And if we can find one, we can find the rest, okay? And then the strand across should be the opposite. So if the strand that we found is the 5' end, then the strand across from it will be the opposite. It will be the 3' end, okay? So remember, DNA is anti-parallel. Which DNA strand below would have the highest melting point, 1 or 2? Well, the correct answer is 2. And the reasoning is because technically this DNA sequence, number 2, has more hydrogen bonds, therefore is a stronger DNA molecule, which therefore would have a higher melting point. So you have a lot of guanine and cytosines in here. Number 2 has more than that of number 1. Okay, so I would say number 2 has the highest melting point. So let's talk about DNA and how we sequence DNA and how we can compare different DNA samples together, because that's important. Because we want to compare DNA sequences sometimes because we want to see how closely we are related to other organisms, or like for evolutionary purposes, or we can see how closely we are related to another human. For instance, what if I didn't know who my father was? It would be easy to tell with a couple of DNA samples, okay? And we can do that by gel electrophoresis. Gel electrophoresis was the first way for us to really analyze and sequence multiple different samples of DNA. However, we use the computer now, and the computer does a lot of our work for us. Now, we do still use different types of gel electrophoresis with the same concept and a lot of research purposes in the lab. But you just wouldn't see that necessarily in a lab where they were doing paternity testing or crime scene necessarily. It would be mostly done by computer because humans make mistakes and computers make mistakes too, but rarely. All right, so the whole reasoning behind gel electrophoresis is this, okay? Down here we have our gels, okay? The gel is used because A, it can withstand an electrical current, because we're going to throw in an electrical current here, okay? And it acts as kind of a sieve or a sifter or a colander, if you will, okay? So because of the gel, large pieces of DNA will stay further towards the wells where we put our DNA in, and smaller pieces of DNA will come towards our positive end. DNA is negatively charged because of the oxygen on the phosphate, on your sugar phosphate backbone. It has a negative charge because it only has one bond and it really wants 2. So that creates a slight negative charge for DNA. So it's going to be really once we turn on the electricity, it's going to be really attracted to this positive charge down here. But then the gel only allows certain small pieces, okay, the smaller pieces, to come more towards the positive end. Because they're smaller, they can fit through that gel easier. Now, what does this all have to do with DNA and sequencing? Well, the different pieces of DNA and their size, it's kind of broken apart, your DNA strands are, by an enzyme called an exonuclease, and it's done so by a specific sequence, right? So where I might have a really large chunk of DNA, somebody that's not closely related to me would have a really small one, okay? So for here, when we're finished running our gel, we want to look for similarities in what we call these little lines. We call them bands. And if we see a lot of similarities within these bands, within each sample, okay, we can say that that organism or that person or whatever we're comparing is more closely related than the others, okay? For instance, let's say we ran a gel on my dad's DNA, my DNA, and my two younger brothers' DNA. Technically, I'm 50% of my dad. I should have 50% of these bands that are the same or at the same length, right? My brothers should also have that. They might not be the same bands as me that we have in common, but they should have at least 50% of their bands in common, theoretically speaking. Okay, so we're looking for similarities here to correlate, hey, how closely related are we with this other organism, right, for evolutionary purposes? Or how closely related are we with this other human, okay, for societal purposes? Okay, so it's actually really interesting. So this is our last question. Does DNA have a positive or negative charge, and why? DNA has a negative charge because of that oxygen on the phosphate group, okay? These are some really good, helpful videos. I really encourage you, if you're still struggling with the 5' and the 3' and all of that brouhaha, please watch this Amoeba Sisters DNA structure video. They go over cars in the garage, apples in the tree, just like we did. So it's super beneficial. If you're interested in learning a little bit more about gel electrophoresis, you can also watch the Amoeba Sisters video on that as well. This base pairing video, this is actually, I believe, either Watson or Crick, I can't remember who it is, as an older man, kind of discussing his thought process on how he figured out how the two bases, that complementary base pairing, so A always binding with T and G always binding with C. So very, very, very cool. I very much encourage you to watch those. And until next time, bye. Have a great day.